And on now to our 7th student speaker. The next one is Hane Ryu. Hane is from the Pharmaceutical Sciences and Pharmacogenomics program, also known as PSPG. She has 2 advisors, Nadav Ahitov and Katie Pollard, and her talk is titled, What Makes Us Human? Hane.
You probably already knew that chimpanzees are our closest living relatives in the animal kingdom. But it's easy to forget just how much we have in common. They have complex social hierarchies, they invent new tools, they teach those skills to their offspring, they even have a sense of humor. But let's not kid ourselves. We're sitting in a room with electric lights, typing on our computers, expressing ourselves in language. So for all of our similarities, there are clearly some dramatic differences between humans and chimps. So how do we pinpoint the molecular changes in our biology that make us uniquely human? One way to do this is to compare our DNA to the DNA of other species. When you compare human and chimp DNA, the humbling truth is that chimps are nearly 99% identical to humans. Although 99% is a lot in common, that still means that of the 3 billion bases that make up the human genome, about 15 million of them are different, which is a vast territory to search. So how do we narrow down the DNA changes that matter most? And how do we link those DNA changes back to our physical and cognitive differences? My research sets out to answer these questions. So we knew that between humans and chimps, our genes, which encode for protein, are nearly identical. And most DNA changes don't happen in these genes. So if you can imagine our genes and protein building blocks as Lego pieces, this suggests that you can make a human and a chimp with the same Lego pieces, but with different instructions. I searched for these instructions in the DNA outside of our genes that's seen as the dark matter of the genome, which is known to contain hidden DNA switches that turn genes on and off. In order to find DNA switches that are human-specific, we compared the human genome to the genomes of other vertebrates to find these DNA switches that have been kept the same across species, but have a lot of human changes. We call these recently evolved DNA switches with human changes human accelerator regions, or HARs. I developed a high-throughput genomic technology to test how well all HARs turn on their target gene in human and chimp neurons grown in a dish. I was able to narrow down our search from those 15 million changes in human to less than 1,000 across 200 HARs that turn genes on in neurons. These HARs turn on genes that help expand the forebrain and enable language. My research not only sheds light on the constellation of DNA switches hiding in the dark matter of our genome, but also takes us a step forward, understanding how very few DNA changes make us uniquely human. Thank you.